The next type of nanomedicines are those that are based on carbon-based particles, of which there are very different or various types as shown here on the right-hand side. We begin with carbon nanodiamonds on A. Carbon nanodiamonds were first discovered in 1961 as a byproduct of explosions. They started being investigated as nanomaterials in 1995. They typically are less than 10 nanometers in size and they have a diamond crystal structure. What that means is that the carbons are bonded to one another through sp3 hybridization, and that means that each carbon is bonded to 4 other carbons through single bonds, and you can see that that repeats over the whole structure of the system. They are the hardest minerals available. They're also great electrical insulators and excellent thermal conductors. On B, we have graphene. Graphene is a carbon-based material in which the carbon atoms are bonded through sp2 hybridization. What that means is that, What that means is that each of the carbons are bonded to 3 other carbons. 2 of the bonds are single bonds, and then the remaining bond is a double bond. That structure provides them with unique properties such as excellent electrical conductance because of the fact that the electrons are delocalized because of the resonance structure that you have here, or the alternating double bond. So electrons are basically free to move throughout the structure, and that's what allows them to be great electrical conductors. On the other hand, the graphite or graphene is also a great thermal insulator, and it's also a soft mineral which enables it to be used as a good lubricant. From there, let's go ahead and skip to D, where we have fullerenes. Fullerenes are also known as buckyballs, and the discovery of buckyballs earned the Nobel Prize in Chemistry in 1996. I would say that this kind of structure has applicability in drug delivery systems because basically what you're forming is a cavity where you could potentially localize other kind of molecules to be able to be targeted for delivery somewhere else. There are similar structures here. So fullerenes or buckyballs have 60 carbons, and so they're known as C60. Here on E, we have a C540, and here a C70, where the number represents the number of carbons that are involved in the making of that structure. From here, on G, what we have is actually amorphous carbon. And finally, on H, we have the representation of single-walled carbon nanotubes, which we will discuss next. Now, in terms of the production method, the production methods are listed here. And they basically include detonation or explosions, arc discharge, laser ablation, and chemical vapor deposition. Detonation is typically utilized for the preparation of nanodiamonds. And I should say before I move on, if you look at all of the methods, they actually all have something unique or something in common, and that is that they require or they involve the heating of the carbon precursors to very high temperature. In detonation, obviously, the explosion is the source of the, not only the source of the carbon, but also the source of the high temperatures, leading to the formation of the nanodiamonds. In this case, the explosives themselves are the source of the carbon typically. If we go to arc discharge and laser ablation, in those cases, basically a carbon gas or carbon gas cloud is formed by either passing a current through carbon electrode or by using a laser to heat a solid substrate in order to reach to very high temperatures in the order of 3,000 to 4,000 Celsius. Then the carbon condenses, producing the carbon nanostructure. And you can utilize different kind of catalysts in those reactions in order to basically provide the shape to the material. Let's see, in arc discharge, you're actually able to modulate the pressure of the gas or the current in order to control the shape of the resulting nanostructure. And I think I'll leave it at that. And then chemical vapor deposition, or CVD, has been used for the formation of nanodiamonds as well as carbon nanotubes. For nanodiamonds, it typically results in the production of a film as opposed to a freestanding particles. Let's see what else. and the reaction typically occurs at about 1,000 degrees Celsius. Now, one thing that I have here that is not really in terms of production, but more of the modification that typically is done for the use of these carbon-based materials in biomedical applications is the fact that because they're carbon-based, carbon only really, they tend to be very, very hydrophobic. And so the applications can be minimal in biomedicine unless they are modified. And typically the treatment is exposure to strong acids. That acid treatment can actually provide carboxylic acid functionality to many parts of the molecule, of the macromolecule, I should say, and that provides it with water solubility. All right, in terms of carbon nanotubes, there are several different types. You can think about carbon nanotubes as basically sheets of graphene that have been folded. And so they're going to contain kind of the same properties, the same sp2 hybridization that I mentioned before. If you have a single wall carbon nanotube, that means that you have a single sheet that has been folded versus multi-wall carbon nanotube where obviously you have more than one sheet. And then we could have also multi-wall carbon nanotube, which is what's shown here, versus double-wall carbon nanotube. These nanotubes can be semiconducting or metallic depending on the orientation of the sheet. I'll go ahead and say that there's three different types of how these materials can fold. There's the armchair configuration, which can lead to a metallic kind of property in terms of the conductance. There's a zigzag configuration, which is semiconductor, and then there's a chiral configuration. And so I encourage you guys to look more into this. I'm not gonna go into details in this area. And here at the bottom right, what I'm showing is the modification of the properties of that carbon nanotube. As I mentioned, we can utilize strong acids such as sulfuric acid and nitric acid in order to provide carboxylic acid functionality, which would increase the compatibility of that material with water. And let's just leave it at that. All right, so the applications of carbon-based materials are many in biomedicine. Of course, we're going to be utilizing the unique properties of these materials, which include the high strength. For example, carbon nanotubes are 100 times stronger than steel. The electrical properties are also going to be very important for the applications. The fact that some of them can be conductive, some of them can be semiconductive, and sometimes the electrical conductivity might be dependent on the direction that we are talking about. The optical characteristics can also be important in terms of the ability of some of these materials to absorb light. Applications include in molecular separations, in biomolecule delivery. So for biomolecule delivery itself, there's different ways to go about it. As is shown here in this graphic, you could utilize one of these carbon nanomaterials to actually load other agents within. So for example, contrast agents or therapeutic agents. That would be an example of biomolecule delivery. In addition to that, we could actually absorb therapeutic agents or biomolecules to the surface of that carbon nanomaterial, as is shown here, with either a protein, I think it's a protein or a nucleic acid that is adsorbed onto the surface. And so biomolecule delivery, of course, also goes with therapeutic treatment in the fact that we can deliver a gene or some kind of nucleotide that has therapeutic abilities, or a drug, small molecule or protein even, that can actually utilize the ability of the carbon nanomaterial to penetrate to the membranes of cells. And the reason for this ability is the hydrophobicity. So the high hydrophobicity of that carbon nanomaterial can enable them to go through the cell membrane without actually having to undergo endocytosis. So we have talked about endocytosis in the past, which is where the nanomaterial might be engulfed and taken in through the endosomal route, which then leads to the lysosomes. But in this case, it can actually penetrate through that phospholipid layer. And so that's something that is unique. In terms of another therapeutic modality is actually the fact that these carbon nanomaterials can generate reactive oxygen species, and that can actually lead to cell damage. So that would be something that you could utilize as a cancer therapeutic, for example. Similarly, in terms of imaging, we can either utilize the carbon nanomaterial itself to provide the contrast. So, for example, carbon nanomaterials can have strong Raman scattering signal, and so that can be utilized for Raman imaging directly. Also, these carbon nanomaterials exhibit band gap fluorescence, and so that could be utilized as well. But not only that, we could also utilize that carbon nanomaterial to load a contrast agent, such as a fluorophore or some other molecule, or maybe even a ferromagnetic or what do you call it, a paramagnetic nanoparticle in order to provide contrast. And then finally, the conductive properties can be utilized for nerve regeneration scaffolds. For example, if you're trying to do tissue engineering for the specific application of nerve regeneration, your scaffold actually has to provide the capability of conductivity because the cells require electrical signals to be able to differentiate into the right tissue. Similarly, you could utilize carbon nanomaterials as electrodes for neural prosthesis and as biosensors, of course. All right, the next type of materials that we have are polymeric micelles. These are self-assembled structures that are prepared from amphiphilic polymers, as we have already mentioned. These amphiphilic polymers are typically going to be diblock copolymers, where we have one block that is hydrophobic, one block that is hydrophilic. The first application of micelles to drug delivery was led by Kataoka and Okano in the 1990s, and this was a polyaspartic acid polyethylene glycol system. Other key players in this area are Vladimir Torchilin, who I mentioned in a previous video, in that he works on liposomes significantly. And then Dennis Disher at the University of Pennsylvania, he works actually on worm micelles, where the structure is not actually this kind of spherical self-assembly system, but where this would actually form kind of a cylinder, if you would. So continuing through the back and to the front, basically, so worm micelles. The characteristics of these micelles include that the morphology can be spherical, rods or worm-like vesicles or lamella, and the sphericals are the most widely studied, and these are the ones that we have talked about the most in this class, of course. They have a narrow size, which tends to be 10 to 50 nanometers, maybe up to 100 nanometers, and as we mentioned, that is a self-assembly process. This is a reversible process, I should mention, so this should in fact have a double-sided arrow, in that they can go back into solution. As you see here, the self-assembly occurs above the CMC or the CMT. CMC is the critical micellar concentration, CMT is the critical micellar temperature. So the assembly occurs above those two. So of course, if the temperature goes below the CMT, or if the concentration of the polymer goes below the CMC, there could be this assembly process going back into the solution phase. And so that could be something that could be utilized as a way to release the cargo that is encapsulated, which is going to be a hydrophobic molecule, of course. Or it could be something that we would have to mitigate against to ensure that it doesn't happen at the wrong location or at the wrong time before the need for that carrier before the carrier is utilized. So yes, micelles are dynamic structures. They are self-assembled, where the assembly is driven by a decrease in the free energy resulting from segregation of hydrophobic segments from water, and is, of course, regulated by the associated entropy gain that is gained by releasing the water that was initially bound. The self-assembly occurs above the critical micellar concentration, or the CMT, and this CMC and CMT can be determined by spectroscopic assays. In those assays, basically, we would utilize a dye, let's say a fluorescent dye, whose spectra is going to change according to the polarity of the environment. One of the dyes that is used for this method is Nile red. Nile red is a molecule that changes its fluorescence. So it actually fluoresces when it is embedded in a hydrophobic environment, but when it goes into a hydrophilic environment, its fluorescence is quenched. So what we can do is that we can put Nile red in the presence of that block copolymer. We allow the micelles to form, And because Nile red is hydrophobic, it would want to load in the core of the micelles. It would preferentially go to that location. And so in that case, we would see fluorescence. But then if we change the concentration or change the temperature to go below the CMT or below the CMC, we can then see that the fluorescence from Nile red would be gone because the micelles would have been disrupted, and now the Nile red is going to find itself in a hydrophilic environment where it is not fluorescent. So typically what we would do would be to investigate the fluorescence here on my y-axis as a function of the concentration of the polymer. And typically it's going to be in a log scale. So this is the log of the concentration of the polymer. And we would carry out the fluorescence measurements, and what we would see is that initially at the low concentrations, we're going to see no fluorescence, no fluorescence, no fluorescence, and then all of a sudden, we're going to see a sharp increase in the fluorescence. We then are going to use a kind of line fitting to look, sorry, that's supposed to be a line. Well, I have no choice but to try to draw a line. All right, you guys imagine a line here at the bottom and then a separate line that is drawn with the slope of this rising fluorescence. And where those two lines meet, that would be considered the concentration of the CMC. We can similarly do something like this for temperature, where we start changing the temperature and then finding at what point the fluorescence actually starts increasing, which means that we would have found the CMT. Another method of investigating the CMC and the CMT is dynamic light scattering, which some of you have had the chance to utilize. And in this case, what we would be looking is the fact that initially scattering would be very minimal because of the size of the polymers in solution would be very small. But once the polymers self-assemble into a nanostructure, light scattering would be would increase significantly because larger particles scatter much more light, and that would basically show up as an increase in the intensity of light scattered or an actual reading of a size of a particle. And then microscopic techniques could also be utilized, including atomic force microscopy and electron microscopy, although they can be a little bit tricky because of the fact that they typically are not looking at the material in its hydrated form. So you might have some false positives where the polymer might just have dried in that form without actually it having been a micelle. All right. The dynamic behavior can be a benefit or a concern for biomedical applications, as I mentioned. So if, for example, the micelle is diluted because of injection into the blood to a concentration that is lower than the critical micellar concentration, then that would lead to immediate disassembly. And of course, that would kind of prevent the micelle from actually being able to target the disease location. On the other hand, we can trigger the release by utilizing changes in pH or temperature or ultrasound or even light in order to be able to trigger drug release on demand. And so that would be something that is an interesting application. The next type of nanomaterials are going to be dendrimers. These dendrimers, as we mentioned, are highly branched, three-dimensional macromolecules, although they tend to be very small in size, usually less than 10 nanometers, and have very high monodispersity and high degree of functionality. They are very monodispersed because they are generated through chemical reactions, controlled chemical reactions, as opposed to a self-assembly process, for example, or an emulsion process. They have high functionality in that each of the terminal groups here would typically be a functional group, like an amino or a carboxylic acid. And that can be of benefit because it enables you to then do chemistry to attach other agents to it, like a targeting agent or a drug. But it can also be problematic. It can lead to significant cytotoxicity. They're characterized by layers between focal points, which are called generations. So this is basically our first generation. Then we go to the second generation here, and then the third generation, and so on and so forth. This particular example is a fourth generation dendrimer. These dendrimers were introduced in the 1970s, and as I mentioned, drugs can be introduced either in the void spaces of the dendrimer or by attaching them to the surface groups. And there are many different architectures that have been published. Another problem with this is that the synthesis of them is highly difficult. So it's not a material that would be readily adapted into clinical use necessarily. And as I mentioned, there have been some issues with cytotoxicity or a lack of biocompatibility because of the high functionality. In terms of the characterization of nanomedicines, we're going to be interested in the properties such as the size, the morphology, the shape, the chemical composition, and the tendency to aggregation. And to study those, the characterization techniques are listed here. They typically include electron microscopy methods. And it would also include elemental analysis for composition, atomic force microscopy to look at the size and morphology or topology of that material on a surface, dynamic light scattering for looking at the size of these particles in suspension. Zeta potential would look at the charge on the surface of those particles, X-ray photoelectronic spectroscopy, which would look at the chemical composition on the surface, and nuclear magnetic resonance, which would typically be looking at the chemical composition as well. Let's see. A couple of things that I wanted to mention here. So EDX or EDXS or EDAX, they're all the same technique, and they're energy dispersive X-ray spectroscopy. And this is going to be looking basically at the elemental composition. Typically, you're going to find these EDAX systems attached to an electron microscope. In our case, we actually have an SEM that has EDAX capability. And so what you can do is that you can go and find the material that you're interested, or even the location on the material that you're interested, and then switch to do X-ray spectroscopy to look at the elemental analysis. And so that can be utilized to look at very complicated nanomedicines that might include maybe gold together with a polymer system, or gold with a biomolecule, and you can actually be able to see if the assembly actually ended up in the composition that you expected. Let's see, what else can I say? In terms of the caveats of electron microscopy techniques, first of all, typically they generally require dehydration of the material, as well as coating in some cases, and that can alter the properties of the particles when you're investigating them. I mentioned coating, that's typically done through sputter coating, and that's typically reserved only to the materials that are non-conductive. And the reason for that is that if you were to image these materials like hydrocarbon or carbon-based materials that are not conductive in the absence of coating, then you might not be able to actually see them at all, or it might generate arcing, which basically disrupts the image. On the other hand, if you coat the material, then the composition is going to be different on the surface, of course, and so you would not be able to use EDAX. Let's see what else. In terms of atomic force microscopy, AFM cannot only find the size and shape, but can also provide us with some mechanical properties such as the elastic modulus or particle adhesion and deformation. So that's something that is of added information that is important. In terms of dynamic light scattering, it works by measuring the particle size or by estimating the particle size as a function of the light scattering intensity. Light scattering is a function of the scattering angle, the particle size, and the refractive index of the particles and of the medium. It is limited to the study of nanoparticles that are in suspension and that have a low narrow distribution of sizes. If you have a very polydispersed system, you're not going to get very valuable information from dynamic light scattering. Also, dynamic light scattering assumes a spherical particle, and so if your particle has a different geometry, then the values that you obtain are not necessarily accurate. Having said that, many companies have been trying to modify the algorithms to enable the characterization of those materials that are not spherical, and so that would be something that you could look into if you are interested in that. One last thing is that dynamic light scattering requires a significant difference in the refractive index between the nanoparticles and the medium. Otherwise, the light scattering signal will be very low. All right, zeta potential, as I mentioned, is going to be looking at the charge on the surface of your particles. It basically, I should say that that charge is going to be important in that, first of all, it enables you to determine whether your particle is going to be stable in suspension or not. So if a particle has a lot of charge, whether that's negative or positive, the interparticle repulsion is going to maintain that particle in a suspended form. Basically, it's going to prevent aggregation. On the other hand, you could also utilize that zeta potential measurement to be able to investigate or confirm the modification of your nanoparticle, the surface modification of your particle. For example, if you are functionalizing your particle with a nucleic acid or something like that, then you could look at the change in the zeta potential, going from maybe a positively charged particle and then going into a negatively charged particle once DNA has been absorbed on the surface or something like that. And the last thing that I also want to mention is that the zeta potential is important when you start thinking about the ability of those particles to interact with cells. So cells are going to have a negatively charged cell membrane, and so negative particles are typically going to have a little bit of a harder time getting close to the cells. And so you might want to actually investigate how does changing the particle charge affect the ability of those particles to enter the cell, if that's what you're trying to do. All right, I will leave it at that. Now, in terms of the applications of nanomedicines, of course, we have already talked about drug delivery. In terms of the nanoparticle acting as a carrier for the delivery of either small molecules, nucleic acids, or proteins. In terms of detection, the particles could be utilized for biomedical imaging, where the particle itself could act as the contrast agent, for example, with the superparamagnetic iron oxide nanoparticles, or the particle could be a carrier for an imaging agent, such as a fluorescent molecule or a gadolinium chelate, for example, for MRI as well, etc. And then the nanoparticle could also be utilized for diagnosis or monitoring in the form of a biosensor, for example, where the nanoparticle could act as a material to separate the biomarker of interest. For example, again, the superparamagnetic iron oxide nanoparticles or other systems. The idea also of utilizing nanomedicines is that they are known to preferentially locate in tumors, and so cancer drug delivery is one of the main areas for nanomedicines. The idea would be that these nanomedicines could be injected systemically through intravenous injection, and the particles would circulate through the body and be able to preferentially accumulate at the tumor site. Once at the tumor site, then they could impart their function, whether that's enabling us to image the location of the particles and therefore the location of the tumor, or release the therapeutic cargo locally, or kill the cancer cells if the particles themselves are the therapeutic agent, with the end product being a cured patient. Of course, that's very idealistic, and there have been papers reporting that even though we are very good at targeting nanoparticles to tumors in mice models, it doesn't necessarily translate directly to clinical applications in humans, and so there's still a lot of work to be done to make this be a reality. This page lists a number of other roles of nanocarriers, and I'll let you guys read those on your own. Let's move on to some of the nanocarrier properties that are desired. So one of the things that we really want is for the nanoparticle to have a long lifetime in the circulation. This way, once the nanoparticle is injected into systemically through the circulation, then it would have enough time to be able to reach the location of interest, which in the case of cancer would be the tumor site. In order to be able to do that, in order to have a long lifetime, it would have to avoid what's known as the reticuloendothelial system, or RES, which is also known as the mononuclear phagocyte system. This is a part of the immune system, and it involves the monocytes and macrophages that are part of the spleen, and the cover cells of the liver. These cells are going to destroy foreign material that has already been tagged through a process known as opsonization, which is basically the absorption of certain proteins known as opsonins onto the surface of the material. After the material is opsonized or tagged, basically these cells can actually identify it and phagocytize it or remove it from the circulation. And so what we want to do is to avoid this, avoid recognition from the immune system. And as we have already mentioned, one of the ways to do that is to functionalize our particles with a hydrophilic polymer, such as polyethylene glycol, which is going to delay protein absorption or the opsonization process, and therefore is going to delay the recognition by the immune system. Pegylation has been shown effectively to increase the circulation time by orders of magnitude, from maybe minutes to days for certain nanocarriers. The other thing that we can do to avoid the reticuloendothelial system is to narrow the size of our particles to be in the range of 10 to 200 nanometers, or I would say 50 to 100 nanometers optimally. Less than 10 nanometers, then the particle would be able to escape through renal filtration, so the particle would have a short time in the circulation. Or alternatively, if the particle is too large, then the particle would be taken away, typically much faster, by this reticuloendothelial system. Now, I mentioned that in the case of cancer, particles would have the ability to preferentially locate at the tumor site. And the reason for that is this passive targeting mechanism known as the enhanced permeability and retention effect, or EPR. Basically, what happens is that as tumors grow, the tumors or the cells associated with the tumor are going to require more oxygen being delivered, because the cells start kind of not migrating, but they start proliferating, and the existing blood vessels are no longer able to supply blood to all of the tumor cells. And so once the tumor reaches a critical size, typically in the order of 4 millimeters in size, then the cells start secreting a number of growth factors that lead to the formation of new blood vessels. That process is known as angiogenesis, or the formation of new blood vessels. The angiogenesis that is associated with tumor growth actually leads to the formation of very immature or architecturally deformed microvessels, which have very high permeability. And that's what's shown here in this picture. Basically, on the left is a blood vessel of a normal tissue, where the cells that line that vessel, or the endothelial cells that line that vessel, are close to one another, have tight junctions in between. On the other hand, in the blood vessel that supplies the blood or supplies nutrients to the tumor, because that's a new blood vessel that is immature and is improper in its architecture, you're going to have large fenestrations or large spaces in between the endothelial cells, leading to high permeability. And we can utilize that in nanomedicine by utilizing nanocarriers that are too big to be able to permeate through the normal endothelium, but that are able to go through these large gaps in the tumor blood vessels, leading to accumulation at this site. So typically nanocarriers that are in the order of 20 to 200 nanometers can extravasate or leave from the blood vessel into the interstitial space of the tumors. On the other hand, the tumors also have a poor lymphatic system, where the lymphatic system is basically the vessels that take the extracellular fluid from the tissues back toward the heart. And tumors also don't have a suitable or a working lymphatic system. So you actually end up building pressure at the tumors because you have high exit of blood and nanocarriers in this case, but you don't have removal of that fluid from the location. And so you end up with a high pressure, which can actually diminish the ability of these nanocarriers to stay in this location. On the bottom, what I am trying to show here is an actual experiment showing the ability of a tumor blood vessel on the left to enable the extravasation of liposomes. These are pegylated liposomes that have a size of 126 nanometers. And in the tumor microvasculature, you can observe that we have the whitish is the liposome. We can observe that the liposomes have been able to extravasate from the blood vessels out to the surrounding tissue. On the other hand, on the right-hand side, we have also polyethylene glycol liposomes that have about the same size, 128 nanometers in this case, and we can see that they remain inside of the healthy blood vessels. They are not able to extravasate. So that's kind of proof of this EPR effect, the enhanced permeability and the retention effect that could be utilized for tumor targeting, and that is utilized for tumor targeting for nanomedicines. This is a skeletonized two-dimensional image of the vascular network of normal arteries versus normal capillaries versus the blood vessels associated with an adenocarcinoma or a tumor. What I wanted to show here is the irregular architecture that basically is just to show you the difference in the architecture and which is part of the enhanced permeability and retention effect. We have irregularly shaped, we have dilated and tortuous tumor blood vessels. And then on the next slide, I wanted to show you, this is what's called a corrosion casting system of a microvascular system. And basically the way that they do this is that they inject a resin through the blood vessels, they cure that resin, and then they dissolve the surrounding tissue by utilizing a corrosive chemical such as potassium hydroxide. And so what you're left with is the three-dimensional architecture of the blood vessels that you can then investigate utilizing electron microscopy, for example, in this case. And what they're showing here on the top left, this is a normal microvessel. These two are the same normal vessels, where B is just a higher magnification of the same. And then on the bottom, what we have is adenocarcinoma. What they're trying to show here is that the vessels here, the vessels tend to have the same diameter throughout. So the capillaries would have the same diameter throughout. Well, here we have different diameter of capillaries. Some of them are large, some of them are very, very small. And then we also have basically endings of the capillaries that don't continue to anywhere else. And those are circled right here. So they're called blind-ended vessels, for example. Again, showing that the architecture of the blood vessels supplying the tumors is very different. And that's one of the reasons why we can use them for targeted drug delivery to tumors. One additional example here is scanning electron microscopy showing the luminal surface of a normal blood vessel. So it's basically showing the endothelial cells, which in this case you can see this is basically one cell, this would be another cell. We can see a tight endothelial junction. While on the right hand side we can see that there are gaps in between the cells, or that the junctions are actually very they leave spaces behind, they leave fenestrations behind. For example, this is one cell. You can see the junctions here. These are kind of the processes from the cells reaching to the other cell next to it, but leaving a gap in the middle. All right, the next thing is passively target nanocarriers. This is just a list of nanocarriers that are either approved or that were in clinical trials at this point in 2010. This might have changed in the past 10 years, but actually I don't see any that I know that are approved other than the ones that are already there. So I don't think there's any new ones actually. In this slide, I just wanted to point a few of them, specifically the ones that are approved, such as Abraxane. Abraxane is an albumin type nanoparticle, where albumin is the protein or a protein that is present in your blood in very high concentrations. So it's basically utilizing a natural protein that your body is used to, but this protein is actually cross-linked into the form of a nanoparticle, and that nanoparticle is loaded with doxorubicin, which is with paclitaxel, which is a chemotherapeutic agent that acts by preventing the cells from moving and from replicating. It basically polymerizes their actin filaments, preventing the cells from replicating. This particle is approved for metastatic breast cancer. Another set of systems that I wanted to mention, or that we have already mentioned actually, is Doxil, which is the liposomal doxorubicin that is pegylated. And Myoset is a similar liposome system, but I believe this one is not pegylated. In general, what these nanocarriers that are approved have shown is that they are similarly effective to the chemotherapeutic agents by themselves, but they tend to have lower side effects. For example, Doxil has been shown to have less effect on the heart because doxorubicin is a drug that is known to cause cardiotoxicity. So Doxil has been shown to have less of an effect in the heart, but still causing significant other side effects. So even though these are better, a little bit better than the drugs by themselves, still it has not been the magic bullet that we envisioned nanomedicines to be. So there's still a lot of work to be done to enabling or to gain a better understanding of how these nanocarriers could actually live to their potential. And one of the things that we are investigating nowadays is whether we can use nanocarriers to actually recruit the immune system to fight the cancers, as opposed to using the nanocarrier to kill the cells. And one of the reasons for this is that nanocarriers actually, it's very easily for them to be phagocytized, as we mentioned. And so a lot of them, they end up being taken in by this reticuloendothelial system and interacting with immune cells. So could we actually utilize that as a way to now actually make them useful and make them and be able to fight cancer in a different way? We mentioned the passive accumulation of nanocarriers to the tumors utilizing the enhanced permeability and retention effect. But we can also actively target these nanocarriers utilizing ligands that have high affinity to proteins that are expressed on the tumors. So the idea is that once the nanoparticle has reached the vicinity of the tumor, then the particle would be able to bind directly to the surface of the cells. And to do that, what we would have to do would be to functionalize the surface of the nanoparticle, with a ligand. That ligand could be an antibody, which is a protein, or it could be a nucleic acid aptamer, which is kind of a mimic of an antibody, but is nucleic acid based. Or it could be a small molecule such as a folate or folic acid ligand. Those, again, would have high affinity to a protein on the surface of the cancer cells, which is known as a receptor typically. Those receptors would have to be highly expressed, or what we know as overexpressed, on the surface of the cancer cells, such that we would have some preferential interaction with the cancer cells versus other cells that might be in the vicinity. What else can I tell you? Oh yeah, one other thing that I wanted to show here is that in most cases we're targeting the nanoparticle to receptors on the cancer cells, but there's also investigation into targeting the nanoparticles to the endothelial cells associated with the tumor blood vessel. So can we target the blood vessel itself and maybe prevent the blood vessel from actually working well in that area, and thereby killing the cells in another way? Oh yeah, and one last thing that I wanted to mention here is that sometimes by selecting the right ligand, we could actually encourage receptor-mediated endocytosis into the cell. So endocytosis, again, is the uptaking of these nanocarriers into the cell through endosomes. You could think about it like the engulfing of these nanocarriers into the cell. And so by utilizing the right type of ligand, once the ligand on the nanoparticle binds to the receptor, sometimes that binding event can lead to endocytosis to begin. And so you would basically enable that, you're basically programming the nanoparticle to be able to attach to the cell, and immediately upon attachment, then endocytosis of that particle would occur. So that might be beneficial if the therapeutic agent that you are delivering acts better once it is actually inside of the cells. This slide is showing the type of ligands that can be utilized for active targeting. For active targeting, again, we can utilize antibodies. Antibodies are proteins that are generated from our immune system. I guess I should mention, so our immune system develops antibodies toward foreign materials like virus, like bacteria, for example. And then we can actually collect those antibodies, and utilizing biochemical techniques, we could actually either separate them from a living organism, or we could have them be produced through what are known as hybridoma cells. These hybridoma cells are basically made to be able to synthesize that monoclonal antibody specifically. Now, we could utilize the whole antibody, or we could utilize fragments of the antibody that are still providing the specificity to the target. I should mention that this Y shape of the antibody contains several parts, and just to keep it brief, the parts that are actually active in terms of being selective for the target molecule are going to be on this end right here. And so this is the reason why the antibody fragments that are shown here on the right hand side basically conserve those parts, as opposed to the tail of that antibody that is not needed for recognition. The benefits of using smaller ligands would be that they wouldn't affect as much the size of the nanocarrier itself, because this right here is a big protein that could be in the order of like 7 nanometers or even bigger, maybe 10 nanometers. And so, you know, if the particle is 10 nanometers and the antibody is 10 nanometers, then we might have a size conflict here. Other agents that could be utilized include human transferrin here. Human transferrin is an 80 kilodalton glycoprotein that is found in serum, and its job normally is to transport iron to the cells via a receptor-mediated endocytosis. And so by utilizing the fact that cancer cells tend to overexpress transferrin receptors, then we can utilize this as a targeting agent. Another agent that is utilized significantly is the RGD peptide. We had talked about RGD peptide as being a cell adhesion peptide that cell adhesion molecules on the cells can recognize and attach to. So if we're able to incorporate RGD peptide on the surface of our nanocarrier, that could be utilized to target and bind to the cells. Another one is folic acid right here. Folic acid or folate, folate I should say, is vitamin B9, and the folate receptors are overexpressed in cancer cells. So again, by attaching this to the surface of our nanoparticle, we could potentially enable the active targeting of the particle to the folate receptors on the cells. Other than that, here on the bottom, I have certain sugars or carbohydrates that can also be utilized for targeting. On the right hand side, again, I have the antibodies, but antibody fragments, and then I have aptamers here. Aptamers are going to be nucleic acid mimics of antibodies, and these aptamers are generated through what's known as the Celix process, or systematic evolution of ligands by exponential enrichment. It's a process in which initially you begin with a library of nucleic acids. A library of nucleic acids means that we're going to have a solution that contains hundreds of nucleic acids of random sequence. Those nucleic acids are put in contact with the target, for example, a cancer cell. Or a protein that is known to be overexpressed in a cancer cell. Then some of those sequences from the aptamer library are bound to have some affinity toward the target, simply because of the fact that they might fold in the right geometry to be able to expose or present specific parts of the nucleic acid molecule toward the target. And basically what you would have kind of like a lock and key mechanism where the aptamer is the key, and if it has the right shape and the right chemical nature, then it could potentially bind to the molecule that you are trying to target, right? So once the two are put together, some of them are going to bind to the target. We can then remove the ones that did not bind to the target and elute or collect the nucleic acids that did bind to the target. We can then amplify those sequences and start a new cycle that enables us to basically eventually, after several cycles, we can identify 1 or 2 or 3 nucleic acid sequences that have high affinity for a target. So we're basically selecting for those sequences that have high affinity by starting from a large random pool of nucleic acids. And so yes, we can utilize that aptamer to covalently bind it on the surface of the particle and be able to use it as a targeting agent. And then peptides are other targeting agents that could be utilized. The RGD peptide, for example, is one of them. All right, in terms of active targeting, the receptors that are most typically utilized on the cancer cells are the ones that are listed here. Notice that they are mostly growth factor receptors, which tend to be overexpressed in cancer cells, so EGFR, VEGF, and HER2. In addition to that, we have cell adhesion molecules, such as integrin or the vascular cell adhesion molecule 1, VCAM. We also have transferrin receptor, folate receptor, which I mentioned in the previous slide, and then glycoproteins that can be targeted by utilizing carbohydrates or oligosaccharides. In this slide, I'm listing some of the tumor biomarkers that would be targeted. Again, some of the ones that we had in the previous slide, but also some additional ones, including PSMA or prostate-specific membrane antigen, and then the expression in cancer tissues versus the expression in normal cells, because unfortunately, all of these receptors might be overexpressed in the cancer, but they are also found in normal tissue. So what we're trying to do is to target those that are expressed significantly higher percentages in the cancer versus in the normal cells. That way we would have higher affinity toward the cancer versus the other tissues. All right, the next slide here is basically showing a number of different targeted agents, whether they're liposomes, micelles, nanoparticles, or conjugates. Then the type of ligand that has been used to target each of the receptors. So for the transferrin receptor, this is what we use. For the folate receptor, this is what has been used. For EGFR, we can utilize basically, we can target the receptor, we can utilize an antibody, or we can utilize, another antibody, sorry. And this is the status. So as you can see, there has not been a targeted system approved, and that's still the case in 2020 as far as I know. They're all under research. Some of them are under preclinical investigation. This slide here, again, is another table that includes the FDA nanomedicines that have been approved in general. And this one includes the nanomedicines that we have already talked about, such as Abraxane and Doxil. It also includes some that are actually simply large biomolecules that are considered nanomedicines in this particular case, but maybe not from the materials perspective, where we're looking for a polymer or a gold particle or something like that to be part of the system. One last thing that I wanted to mention is that these nanocarriers can be multifunctional, and we have already said that in a couple of times. Basically, they can combine passive and active targeting. The passive targeting is by utilizing the size of the nanocarrier mostly. The active targeting is by utilizing ligands on the surface of that system, for example, an aptamer, to be able to target specific biomolecules on the cancer cells or on the diseased cells that you're trying to treat. The function of the nanocarrier can be triggered by internal or external stimuli. When I say internal stimuli, I'm talking about different conditions inside of the patient's body. For example, changes in the pH or enzyme expression or temperature or molecular composition of the target diseased tissue. In addition to that, those nanocarriers can act, as I mentioned, as contrast agents for imaging by the inclusion of dyes, which would enable optical imaging, or by the inclusion of imaging isotopes to enable scanning in other modes. They could also incorporate therapeutic agents such as drugs, toxins, nucleic acids, or even therapeutic isotopes. And they can be made to act kind of intelligently to be able to enable targeting and enable drug release on demand when they are needed, etc. All right, from here, I just wanted to talk about a couple of examples. This first one is near-infrared fluorescent nanoparticles for combined MRI and optical imaging. In this particular case, what we're seeing here is a protease cleavable polymer. Protease is a type of enzyme that has the role of cleaving a polypeptide. So this polymer, which is a polypeptide, was being used to be able to bring a couple of things together. So notice that here we have a sulfhydryl group or a thiol group. Remember that we can use thiol groups to enable binding to gold nanoparticles. In this particular case, they were not using gold nanoparticles, but instead magnetic nanoparticles, specifically superparamagnetic iron oxide nanoparticles. And the chemistry of binding to those particles is the same as the chemistry for binding to gold. So we're going to be using this to now attach a nanoparticle here that enables us to do MRI imaging, which is what's shown on the right-hand side. In addition to that, here we have a reactive amine that is utilized for attachment of a fluorescent molecule, and that is going to enable fluorescence imaging to take place. In this particular case, we mentioned that this is protease cleavable. And so what they did is that they showed, one more thing that I need to mention before we continue. So the presence of this nanoparticle here versus the dye that is on this side. If these two are close to one another, the fluorescence from the dye is actually quenched. And so only once the polymer has been cleaved through the action of those proteases will the fluorescence actually be developed. The proteases are overexpressed in tumors, and so we could potentially use these particles to be able to develop fluorescence at the tumor site because of the function of the enzymes. And we can see that here. Upon activation by trypsin, we can see the fluorescence enhancement. And then on the right-hand side, we have MRI imaging of the lymph nodes showing the localization of the superparamagnetic iron oxide nanoparticles, which, as you remember, give you negative contrast. And then on the bottom, this is the same animal. On the bottom, we're going to see the fluorescence imaging, where here we can see the fluorescence being developed. So the location is the same, showing that the location of the nanoparticles, we are in fact seeing fluorescence being developed. All right. In the next nanomedicine, we have a theranostic system. What we have is also a polypeptide right here that has a Cy 5.5. This is a fluorescent molecule. On the right-hand side, we have cysteine. Cysteine is a peptide, or an amino acid, I should say, that contains a thiol group. And so that thiol group would enable us to attach this polypeptide to a gold nanoparticle, in this case, a gold nanorod. So here we see the assembly of the system where we have a gold nanorod that is now functionalized with our peptide, and the fluorophore is represented by this sphere, blue sphere. Initially, this is quenched, again, because of the close proximity of the fluorescent dye to the gold nanoparticle. The gold nanoparticle quenches that fluorescence. Now, once that nanoparticle is in the tumor site, the tumors overexpress these proteolytic enzymes or proteases. One of them is MMP, which is matrix metalloproteinase. And the MMP is then able to cleave this polypeptide, enabling the fluorescent molecule to diffuse away. Once it diffuses away, it is able to fluoresce, so it's basically activated, and so you see the fluorescence development at the site. Here, what we're seeing is near-infrared imaging, fluorescence imaging of mice, and let me see if I remember. Number one is gold nanoparticles, MMP, meaning that we have fluorescence development. Number two is the nanoparticles that have the fluorophore also that are in the presence of an inhibitor. And so what you can see is that in number one, in all cases, we have fluorescence development. On number two, we have an inhibitor of the enzyme, meaning that that cleavage process cannot occur as readily, and we can see significantly lower fluorescence development. So that's a proof of concept just to show that, in fact, the fluorescence that we're seeing is from the development of, or from the activation through the function of the enzymes. In this next one, we have a liposome system that on the surface has two types, or actually three types of functionalization. One of them is just long polyethylene glycol chains that are attached through this pH cleavable bond right here. I highlighted the wrong one. The next one is going to be the short peg spacers or short peg chains that are not pH sensitive, but that do carry this triangle, which represents this is a cell-penetrating peptide. And then the last set is going to be these long polyethylene glycol chains that are also attached to an antibody to enable targeting. Notice that those are also bonded through this pH cleavable bond. And so what would happen is that this nanoparticle could circulate, it could accumulate at the tumor. Once it is at the tumor, tumors tend to have lower pH in the range of about 6, and so the pH cleavable bond would break, enabling the release of the long peg, whose role is just to increase the circulation time in the body, and that would enable at the same time, we could have the targeting to the cell receptors, but then once these are cleaved, what is left is only the particle with the cell adhesion peptide that would then enable the nanoparticle or the liposome to enter into the cells directly. So that's an interesting SMART type particle. This is by the group of Dr. Torchiling, who I mentioned before as well. This one is from the work of my group and Dr. Shannon Wycombe's group as well. In this case, we were trying to develop a nanomedicine for the treatment of liver cancer. And so what we did is that we utilized polylactic coglycolic acid block with a polyethylene glycol block, and we utilized a carboxylic acid as a terminal group on these particles. We also blended this polymer with another polylactic coglycolic acid that had a polyethylene glycol that has a methoxy group, which is not reactive. We then assembled that into a particle system in the presence of doxorubicin, which is a chemotherapeutic agent, and that led to the formation of this type of core shell particle that is similar to a micelle, but it's a more stable particle because of the molecular weight of the PLGA portion. This particle again has doxorubicin in the center. We then utilized the carboxylic acids that were left on the surface of the polyethylene glycol to now attach an aptamer, which, as I mentioned, is a nucleic acid-based molecule that has high affinity for a particular target. In this case, the target was actually liver cancer cells. And so now we have covalently attached or covalently decorated the nanoparticle. With those nanoparticles, we investigated the ability to release drug in a controlled manner over a long period of time, in this case, about 40 hours at least of controlled drug delivery. We also investigated the ability of these nanoparticles to target the cancer cells. And I unfortunately did not include the picture here of the cells where we utilized a fluorescent agent to show that the nanoparticles actually did attach to the surface of the cells. But what I do show is the ability of the nanoparticles to kill the cancer cells. So reduced viability means increased cancer killing ability. And what we found is that our nanoparticles with the targeting agent, which is the blue, behaved almost identically to the free drug in solution. So even though we didn't get better results than the drug by itself, still we think that this nanocarrier would be better because it would protect the healthy tissues as opposed to just using a drug solution by utilizing the passive targeting. Oops. Let's just continue and finish up. The next nanomedicine is also from my group, and in this case what we were trying to do was to load a nanoparticle with a fluorescent agent to enable near-infrared fluorescent imaging. So this is a polylactic acid polyethylene glycol block copolymer. We loaded a near-infrared BODIP molecule which can fluoresce in the near-infrared where our bodies are more transparent than in the visible range, of course. And one key thing here is that this dye is so hydrophobic that if you put it in water, it basically not only goes to the surface, but kind of climbs up the vial. It really does not want to be in contact with water. And so it could not be utilized for biomedical purposes unless it was put as part of a nanocarrier. And what you can see here on the second picture is that once we form the nanocarriers, we are able to include that dye or to incorporate that dye in an aqueous suspension. And so, yes, we were able to show that we could obtain near-infrared imaging of our cells. All right, the next agent is also from my group, and this is a nanoparticle that is made from polylactic glycolic acid that is it's in a block, I should say, it's a block copolymer of polylactic glycolic acid with a block of poly-L-lysine. That poly-L-lysine is a peptide that has a lot of amine groups, which would enable us to attach a lot of things to the surface of the particle. We blended that polymer together with polylactic acid, block polyethylene glycol with a carboxylic acid end group. And so once these nanoparticles assemble, what you end up with is a nanoparticle where the hydrophobic regions are going to be in the core, and the hydrophobic regions are this and this. And then all of the other parts are going to be on the surface, meaning the poly-L-lysine and the polyethylene glycol. We then utilize that poly-L-lysine to attach a fluorescent molecule, Alexafluor 750. But now, since we have so many Alexa fluors on the surface, they actually quench each other. And so initially our particle is not fluorescent. But once enzymes are able to cleave this polypeptide, then we get fluorescence development, similar to the work that I showed you a little bit before. And so with this system, we were able to show fluorescence development up to 15-fold in the presence of enzymes versus in the absence of them. And we were able to show in vitro that in fact the fluorescence developed on the cancer cells as a function of time versus cells that were treated with an inhibitor that prevents the enzymes from working, in which case you don't get almost any fluorescence. The last system that I wanted to talk to you about is a nanoparticle that is utilized for photothermal therapy, and this is also work from my group, and I apologize for not putting the reference here. But basically, these are nanoparticles that are used for photothermal therapy. Photothermal therapy is a treatment modality in which the particles themselves absorb light and convert it into heat. And so thereby you could actually kill tumors locally by increasing the temperature above a temperature that would basically end up damaging the cells. To prepare these particles, we use an emulsion process utilizing a monomer. That monomer could be E dot, or it could be this large monomer which we labeled M1. In the case of E dot, they just basically end up forming this P dot polymer structure. And the particles from P dot were in the size range of less than 100 nanometers, as we can see in the TEM, SEM, and dynamic light scattering. The most important part is that these particles are able to absorb light in the near infrared range, which is basically from about 700 to about 1,000 nanometers in wavelength. That is important because in this range, our tissues are mostly transparent. As I mentioned before, the biological chromophores typically absorb in the UV or in the visible range. For example, proteins and DNA absorb in the UV range. In the visible range, we have hemoglobin and oxyhemoglobin from our blood, and in the near infrared, there's very few chromophores that would have any absorption. So by doing that, we can use laser light to excite or to interact with these nanoparticles and not affect the surrounding tissues that would also be irradiated because they would not absorb the light and therefore they would not heat. We then were able to show that these particles in fact did heat up. So we have 3 different concentrations that are, or nanoparticles that are irradiated, and certainly they increase in temperature, so a delta T of 45 degrees Celsius. In reality, all that we need is about 5 degrees Celsius in order to completely kill the cancer cells. We showed that the nanoparticles were cytocompatible, meaning that they did not decrease the viability of the cells at even very high concentrations. So that's a good thing. And we were also able to show that the cells were alive in the presence of the nanoparticles as long as the laser was off. But once the laser was turned on, the cells actually died. And this is a live dead assay that I can tell you about later on. So with that, I'm going to go ahead and finish up this lecture.